right? So we have metazoans. And under metazoans, we'll be talking about invertebrates and vertebrates. Okay, so we are talking about invertebrates and vertebrates. But today's class, we will be mostly talking about protozoans. But before going into protozoans, a little bit more about animal kingdom is how we have been deviated from acellular to unicellular to multicellular is a real change, a massive change, that is from a tiny cell, that is which is almost invisible ones, microscopic ones. From there, this big chordates, that is live on human beings. We are even talking about mammals like whales, elephants. So a tiny microscopic living organisms has become almost like a elephant or a whale. That is, there's so much of tremendous changes that occurred throughout the generations after generation. So we humans or chordates just didn't happen like that. It took so many years for us to change and come to this level. So in this process, where when you're talking about unicellular means single cell. So that single cell was taking care of all the aspects of what they have to do as a living organism. What is the first important point of living organisms? Living organisms is nothing but we have to eat to survive, right? So we are going to eat to survive. Without eating, nobody is going to survive. Human beings, why we work so hard? We work so hard only to earn money so that we can eat. So even one day, if you're not able to eat even one day, what will happen? We cannot survive at all. So we have to eat. We have to earn for that. So in the same way, the microorganisms also keeps moving from one place to another place in search of food. So when the single cell can do so much of work, when the multicellular organisms are growing this big, each cell has specialized itself to become a tissue. That tissue became an organ, and the whole organism came into form. So what are the changes first? So we can talk about respiration. So the respiration, earlier animals, it was just diffusion, right? And later on, you have specialized organs, like first when you're talking about the pisces, so you will be talking about the gills for respiration, and then later on that gills change into lungs, and then you have become a specialized one. So that is one example. Then you can talk about reproductive system. So earlier it was just a hermaphrodites or just single cell that gives rise to, or by binary fission or conjugation or syngamy, there was reproduction. But later on, the proper reproductive system with an egg and a sperm releasing reproductive organs came into being. Then the next important is the foremost one is when they were moving, the movement is you can find in protozoans and the bacteria, it's only by pseudopodia. But later on, that changed into limbs or organs. How? By the presence of the microfibrils and the actin and the myosin development, which became into a muscle, which was able to walk properly. So you can take an example of human beings where we are able to walk just because of the muscles, that is the myosin and the actin proteins that are present in the muscles, which makes us to walk. So that is one more important point on development of animal kingdom. So rest all is the same, like different types of organisms shaped up accordingly so that they can survive in the human in the environment. So for example, pisces, so they have developed the fins for swimming and gills for respiration, because why do they have gills and not the lungs in the fish, leave alone the lung fishes? So when you're talking about the general fishes, they have only gills but no lungs. Can anyone tell me why so? Why the fishes still have gills and not lungs? Any answers? Can I get any answers from the participants?
Using
Because
lungs,
they
we
respire
cannot
to this.
respire in water.
In water. Why so? Why so?
Because they respire through gills.
what is happening in the respiratory?
They can only take dissolved oxygen which is present
Exactly.
in water.
Very good. Very good. So the gills can take up only the dissolved oxygen present in the water, which a lung cannot do. A lung can only take the atmospheric oxygen. So they have been adapted in such a way they can still survive in the water and they can take in the dissolved oxygen in them. And what are the other ones? So the amphibians. Amphibians, they can live in the water and also on the land because of their own specific developments. And they also secrete some slime so that all over the body so that they can escape the prey. Then reptiles. Reptiles, they can crawl on the ground and even without the limbs, they can survive because they have the ability to crawl. Then we have the apes, the ability to fly so that they can see the whole world. That is, with a bird's eye, they are able to see the whole world. That's a beautiful one over there. And then, of course, the mammals. Why mammals? Because they have the mammary glands so that they can take care of the young ones. So they have changed themselves. So from unicellular organisms, you can see how they have completely changed themselves to multicellular organisms, and what are the different time gaps where the unicellular organisms change into a metazoan or a multicellular organisms. So that was a little bit of information on animal kingdom. So we will go into the detail on the unicellular organisms, that is the protozoans. So here we will be talking about the general characters of the protozoans, and we will also talk about the type study of different subclasses of this protozoans. And let me share the screen for you all. 1 second. This is my first class. Can anyone tell me how to share my screen? Like, I have a PowerPoint presentation and then I'm unable to open it.
Ma'am, in the middle, the green one, yes, ma'am, this one, like this only.
But that is not the one I want.
Okay.
I have a PowerPoint presentation on the screen.
Okay, ma'am. In the meeting settings, it will be there, ma'am.
Meeting settings.
In the more one.
More I did, but where is that? I am losing.
There is also option of share screen.
Yeah, I did that share screen. My screen has reduced itself. I'll just take the help.
మేడం అన్ని చేయాలి.
Yeah, can you see the screen now?
Yes, ma'am.
Yeah. Right. So we'll be talking about the general characters and classification of protozoa. So you can see the different um, characters over here. That is Paramecium, Radiolarian, Euglena, and so on. You're able to hear me?
Yes, ma'am. You are audible.
Yes,
Yes, madam.
Fine, fine. So this is the invertebrate phyla and approximate number. I just love this information about how many animals are there in the world, and the phylum Protozoa consists of 50,000 species. Can you see the difference? And the next highest in the environment is the Arthropoda with a number of almost 9 lakh species. So we are having
Please
a
mute your devices.
pardon, ma?
Ma'am, someone mute is on, ma'am.
You're not able to hear me?
No, ma'am.
no, ma'am. Disturbance is there, ma'am.
Ma'am, you can mute them. You can mute them, ma'am.
Yeah,
Ma'am, there is
please.
disturbance, ma'am.
Yeah. Can you all please mute your audio, please, so that others can hear clearly? Thank you. So,
Okay,
and
ma'am.
Arthropoda is at 9 lakh species. So, the environment or the earth has a maximum number of insects. So, we are surrounded by lot of insects. That's the reason why any nook and corner you go, you will find some insect or the other roaming around. So, that is the amount over there. And our protozoan is at 50,000. Yeah, somebody is having a question. Yes, ma'am. What's the question? Okay, so if you have any doubt in this picture, you can just come back and you can also go through this picture over there. So what are the general characters? These are microscopic, eukaryotic, unicellular. Now, can you see the difference? They are unicellular, but also on the eukaryotic. Are you able to understand the difference? Even though they are unicellular, single cell, what is the feature of a eukaryotic? They have the nucleus and a nuclear membrane. So the nucleus is surrounded by a nuclear membrane. That is the reason why they are under eukaryotic. They are solitary or colonial. That is either they are single or they are in the colony form, and they can be motile sometimes, or they can be stationed at one place, and they are phagocytic, heterotrophic animals. Phagocytic. So what exactly is phagocytic? That is, they can absorb their food, or also they can phagocytose the food. That is, they have a amoeba like the pseudopodia like structure which engulfs the food, and they can just consume it completely. So that's a phagocytic. Heterotrophic, anything can be taken, and these are animals without tissue or organs. So that is the reason why we call them as unicellular. So they don't have a proper tissue and organs, and they can have one or more nuclei. So please underline this. Remember that these protozoans can have a single nuclei, or they also can be made up of many nuclei. And as I said, almost 50,000 species are known till date. And who is the person who first observed protozoa? Anton van Leeuwenhoek in 1676 was the first person to observe protozoa. Which one did he observe? He observed the Vorticella, and this Vorticella comes under Celiophora, subphylum Celiophora, and they call them as animalcules. And Gold first coined the term protozoa in 1818. So students, just imagine the scientists then, way back in say 17th century and 19th century, and look at the thing that they have observed. They were not sitting quietly, and for us they have given everything on a platter to know and understand what they are. So I hope each and everyone in the future will become scientists, so that you will also keep searching something new. So study is not just that degree pad liye or then we are okay, we got the marks and we got lost. No, take your studies to the next level so that you can also have so much of knowledge, you can become a great scientist in the future. Fine. Coming back to our topic over here. So they may be free living or symbiotic. So amoeba is a free living. Symbiotic, what do you mean by symbiotic? That is, they can share, symbiosis, sharing the place. They are either in the host and they take the shelter in a host, and they can also give some help to the host, or they can become parasitic. That is why under symbiotic, you have a parasitic one, a mutualistic or commensalist one. So what do you mean by mutualistic? Mutual understanding. So I will take shelter from you, I will give some food for you. That is, if there are some photosynthesis animals, they photosynthesize the food and the food is given to the host. So that is called mutual understanding, give and take policy. And body is symmetrical, that is how you can divide the body. So they're either symmetrical, that is actinopodians example. They can be radially symmetrical, they can be bilaterally symmetrical, or no symmetry at all, asymmetric, that is in amoeba. That is what I mean by exactly symmetry. When you cut a body into two halves, it should be divided into two equal halves. That's called symmetry. So in radial symmetry, that is, for example, a starfish, you're going to cut them radially so that you get, yeah. Any question?
Indo-European language.
Ma'am, page number, please, ma'am.
Yeah.
Page number, book page number.
Book page number. See, I'm not following the book. You just follow what I'm teaching you all.
Page 10, page 11.
I don't have a textbook with me right now. I am talking about protozoans. Classification, general characters and classification.
Chapter second.
Yes,
Yeah,
ma'am.
I finished
Page
unit
number 10,
1.
ma'am.
That is, introduction to animal kingdom is over. Now I'm talking about the second topic, that is protozoans. General character and
It's page number 10.
Okay.
What it? For now, you all just forget about the textbooks. Just follow what I'm teaching.
Yeah.
Later on, you can go back to your textbook and then you can read it.
Okay, ma'am.
Yeah, please mute that sound. Baby's crying somewhere, please. Fine. Okay, so radial symmetry. See, in science, you will have to first understand some important scientific names. If you don't understand the scientific names, you cannot follow your degree. So the textbook will not give you all this in detail. He will only say body is symmetrical, radial symmetry, or bilateral symmetry. But what is symmetry? Some will be knowing, some will not be knowing. So what I'm teaching is for those who don't understand what it is. Am I clear? Okay. So by symmetry, symmetry is when you cut a body into two halves, the two halves should have all the parts in them. So that is what is called radial bilateral symmetry. But all organisms are not bilateral symmetry. Any doubt?
Clear, ma'am.
If you have any doubt, only then you unmute it. Otherwise, please keep the mic under mute so that others are not getting disturbed. Okay, so I hope you have understood symmetry. Now we'll go to locomotion. That is how they move from one place to another. Now we all know that amoeba moves by a pseudopodia. That is pseudo. Pseudo means false. Podia means foot. A false foot is present in them. That is, they produce their own false foot and then they move in the forward direction. So that is a pseudopodia. They can also have a flagella like the euglena. They have a flagella, whip-like structure over there. Or they can also move by cilia or myonemes. It can also be called as some muscle structures, but not exactly muscles, myonemes or cilia. So that small extensions that you find on the paramecium. So that is the cilia. And what is the use of this locomotions? They are able to move from one place to another in search of food. So ultimately, every living organism needs food to survive. Now, what they do and how they eat, that is nutrition. nutrition is either holozoic. What do you mean by holozoic over here? Now, if you can see, this amoeba creates a false foot. So this foot extends around the foot particle over here. The blue dot is a foot particle. The foot extends and surrounds around the foot particle. They engulf the foot particle, and a food vacuole is present. And this food vacuole produces some enzymes, digests the food, and they are assimilated in the body surface. So that is how they are going to eat. So that type of nutrition is called holozoic or holophytic nutrition, or osmotropic is also one and the same. They can also sometimes be saprophytic or parasitic. Parasitic, we all know how they live on the host and destroy the host ultimately. So parasite and mutualistic type of food is different. So parasite is the one that is going to only gain for itself, but the host is going to be destructed eventually. And if you can see in this part, what is happening in the excretion after the food vacuole has been taken inside, and once the food has been digested, the whole food over here is consumed, and ultimately what is happening is the digested food, the undigested and the unwanted food is thrown out by the food vacuole. So this is how the excretion occurs. So nutrition is a holozoic type of nutrition where the digestion is intracellular, and the excretion occurs by diffusion across the general body surface. That is, you can find that the vacuole has been expelled out. And this is a common feature, that is the vacuoles are the common feature of osmoregulation. I hope everybody knows what is osmoregulation. It's a big topic again. So, but as of now, you can just understand that osmoregulation is to maintain the minerals inside the body and maintain the amount of water that is present inside the body. Coming to respiration over here, it's by diffusion through general body surface, and they can be aerobic or anaerobic type of respiration, and anaerobic is usually found in the parasitic forms. And reproduction is through binary fission or multiple fission or plasmotomy. Plasmotomy is the budding of the plasma within the body, or it's also called as budding. So these are the different types. So in amoeba, you can find that's a binary fission, division of the mother cell into two daughter cells. Then the flagellates and the cilia also you can find the binary fission over here. And in Vorticella, you can find the budding type of reproduction over here. So a small bud comes out and chops off. Yeah.
Small bud is formed.
Bud, yeah. So this is a bud, and that is why it's called as budding. So when a bud is formed, they form into a small bud-like structure, and they are chopped off, and they start to live separately, or they are connected to the mother cell itself, and they are joined and they grow separately. And this is plasmotomy. You can find one single cell dividing multiple times, and the plasma cells are being divided. That is, the cytoplasm and the nuclei separates itself, and that is called a plasmotomy kind of reproduction. Now, what is we have completed asexual reproduction. Now coming to sexual reproduction, this can occur by conjugation. So you can see the Paramecium over here, for example. You have a conjugation type of reproduction, or by fusion of gametes. So you can find the exchange of gametes occurs over here, and this type of fusion of gametes is called syngamy. So the name for that is syngamy. Either conjugation or by fusion of gametes is called as syngamy type of reproduction. And one more important feature of this protozoans is they undergo encystment. That is, when the condition is unfavorable, to tide over that unfavorable conditions, the type of these Plasmodium or it can be any protozoan, they form an encystment. That is, they become rounded off, and they have a thick covering around them, so that they can tide over unfavorable condition. And once the favorable condition returns, the cyst is open, and the individual organism comes out, and they are living freely again. And What is another important point? Somatoplasm and germplasm are not differentiated. As I told, these are unicellular organisms and they are not having any specific kind of tissue or organs for them. So the somatoplasm and the germplasm are not differentiated. They are dispersed everywhere in the animal and hence they are immortal. Why you call them as immortal? That is, they don't have death at all. Why you call them as immortal is because they don't have a specific tissue or organ for this particular work. For example, for us, we have different organs for different work. And any one organ is having any destruction or any accident occurs, or if it stops working, then we ultimately die. But here, each and every cell works on the same line. So even if you take out one or two cells, the cell or the protozoan is not going to die. So that is why they are immortals. And for the encystment, you can see over here, so this is a mature trophozoite and which undergoes cyst formation over here. So this cyst is the one, once a favorable condition comes back, the cyst is opened up and the animal comes out of it. So this is a pre-cyst, mature cyst, an intermediate form, and once the favorable condition occurs, the individual comes out of the cyst and they are free living. So that is about the reproduction part. Now coming to, so that was a general characters. So if you are asked a question to talk about the general characters of the protozoans, you're going to talk about all those topics that I've shown in the previous slides. So if you remember all those points, you can just put them on the paper. Now coming to the next side heading, classification of phylum Protozoa. This question is also a favorite question for you all for your examination point of view. They might ask you to define or write a few lines about classification of Protozoa. So you will be needing to write all these points over here. That is, the protozoa is divided into 4 subphyla. Here we'll be talking only up till the class. Okay, so we'll be talking about the phylum, subphylum, superclass, class. So only up till the class, we are going to talk about the classification in this protozoa. So please don't get confused. If you remember this page, you'll be able to write at least 4 or 5 pages on this point. So what are they? So classification is given by Honenberg and a society of protozoologists divided this phyla into 4 subphyla. So all the 4 subphyla are important. So what are the important points we'll be talking about is, the first subphyla is Sarcomastigophora, and this Sarcomastigophora is divided into 3 superclasses over here. And what are the 3 superclasses? We have the Mastigophora, Opalinata, and Sarcoidina. Okay, and that subphylum Sarcomastigophora. Sarcomastigophora is divided into 3 superclasses: Mastigophora, Opalinata, and Sarcoidina. This superclass is again divided into different classes. That is, Mastigophora is divided into 2 classes: Phytomastigophora and Zoomastigophora. So student, under this Sarcomastigophora, Mastigophora, and class Zoomastigophora, this is very important because we are going to talk 2 important disease under this topic under Sarcomastigophora. So remember this Zoomastigophora. So what do you mean by Sarcomastigophora? Sarco means fleshy. That is, the body is made up of fleshy characters. So that is why it's called Sarco. So you can just write on in a book or a notebook. Sarco means fleshy. Mastigo means whip-like structure. The whip, that is a flagellum that you call. So that's called as a whip, W-H-I-P, whip. Sarco, flesh. Mastigo, whip. And phora is they bear, bearing, or they have. Phora is to have or to bear. Okay, it's not that bearing the pain. It's not like that. Bearing is to have it within themselves. So fleshy flagella or fleshy whip-like structure comes under Sarcomastigophora. Understood that point? When you're talking about Sarcomastigophora, I said we are talking about 3 superclasses: Mastigophora, Opalinata, and Sarcoidina. What do you mean by mastigophora? Again, mastigophora. Mastigo means whip, phora means bearing. So they are going to have whip for movement. That is for locomotion. They have this whip for movement. So under class mastigophora, we'll be talking about phytomastigophora and zoomastigophora. Phytomastigo means, phyto means, you all know by the name, they are plant-like animals, plant-like ones, which is having whip-like structures. So what is the example over here in phytomastigophora? The example over here is Euglena. So remember, we saw the picture Euglena over there. Euglena has the nucleus, the macronucleus, the micronucleus, and it also has chlorophyll. So what is the use of a chlorophyll? It helps in photosynthesis. So they are kept under both plant and animal kingdom. That is, they act as both plants and animals. That is because of the presence of the chlorophyll, they are able to produce their own food, and with the help of the food vacuole, they are able to engulf the food by holozoic nutrition. They are going to engulf the food and they are going to have their food. So that is phytomastigophora. Zoomastigophora, zoo means they come under exclusively animals. That is the animal that depend on plants. So that's why they are called as zoomastigophora, or animal which is having whip-like structures. So that is zoomastigophora. Fine. Now, you will be talking about the superclass Opalinata. Opalinata doesn't have a class, so directly the example is Opalina over there. And then you will be talking about Sarcoidina. The Sarcoidina over here, it is made up of 3 classes: Rhizopoda, Phyrophasmida, and Actinopoda. Sarcoidina is having 3 classes: Rhizopoda, Phyrophasmida, and Actinopoda. Sarcoidina. Sarcoidina means, again, they are fleshy-like animals. Sarcoidina. That is why Sarcomastigophora over here, what you call them as Sarcomastigophora. Sarcoidina, they are fleshy ones. Under the fleshy animals, you are having Rhizopoda. Rhizo means root-like foot. Poda means foot, I told you all. Root-like foot is Rhizopoda. And we are going to talk about Phyrophasmida. The Phyrophasmida is the pear shape. Remember the pear-like apples that you get around, the pear shape. So all those animals that is having the pear-shaped ones, they come under Phyrophasmida. And then Actinopoda. Actino means acti means ray-like foot is called Actinopoda. So the examples we'll be talking about in the later pictures. Then coming to Sporozoa, the second subphylum over here, Sporozoa. Sporo means spore-like structures, spore-like animals, zoa. Spores, all the round spores you call them, so that all come under Sporozoa. Under Sporozoa, you have 3 classes: Telospora, Toxoplasmia, and Haplosporia. So these are the 3 classes: Telosporia, Toxoplasmia, and Haplosporia. Again, in the Sporozoa, we'll be talking a little bit about Teleosporia. Teleosporia is the one that we are going to talk about when you're talking about the diseases in the protozoa. Teleo means thick-walled. Teleo means thick. They are made up of very thick-walled spores. So that's why they're called as Telosporias. Why they are made up of thick-walled is, once they attack any host, the digested juices should not kill them. So that is why they are made up of thick shell, and they are not so easily dissolved or killed by the digestive juices of the host. Okay, so under that we'll be talking about Plasmodium. Fine. So I hope you remember what a Plasmodium is. What is the disease that that is caused by the Plasmodium? Anyone? Can anyone tell me what is the disease caused by Plasmodium? Malaria. Yes.
Malaria.
Malaria. Yeah. So that is the reason why I told you. Remember this Teleosporia. We'll be talking a little bit more about that. Then you have Toxoplasma. Example is a Toxoplasma itself. And Haplosporia, you have an example of Haplosporidia. That is the example. So mainly we'll be talking about Pterosporia. Then coming to Nidospora, nido means needle. Needle like spores is Nidospora. And under Nidospora, we are going to talk about two classes, Myxosporidia and Microsporidia. And not much we are going to talk about this Nidosporia and Microsporia. Examples are Myxobolus in Myxosporidia, and Nosema bombycis is the example for Microsporida. And coming to Celiophora, celio means the name itself tells, that is, this animal is made up of cilia. The whole body is covered by cilia. So the class is Ciliata over here. And what are the examples over here in Ciliata? We'll be talking about Paramecium and Vorticella. So this is one more picture I gave you all so that you can understand this classification with the help of the examples that I was talking about over here. So here, when I'm talking about Mastigophora, I told you all the Mastigophora is most important with two classes, Euglena and also Zoomastigophora. And Zoomastigophora, we will be talking about Leishmania and Trypanosoma. So these are the two important protozoans that we'll be talking over here. And then under Rhizopoda, we'll be talking a little bit of type study, that is Lphdm. And of course, we'll be talking a little bit of amoeba, entamoeba histolytica. And when you're talking about Telosporia, we'll be talking about Plasmodium. And yeah, so these are the four important ones we'll be talking about. That is Leishmania, Trypanosoma. Then we'll talk about Lphdm, and we'll be talking about Plasmodium. Okay. So first, coming to the first superclass, Mastigophora. Mastigophora, as I told you, mast means whip-like structures is Mastigophora, and the body of the animal, animals belonging to this superclass is covered by pellicle. So if you can see over here, so they are covered by a pellicle, and locomotory organelles are flagella, and reproduction is by binary fission. We talked about how they have a fission over there. Under this, you'll be talking about two classes, Phytomastigophora. What are the important characters in Phytomastigophora? Phyto means plant, whip bearing. So this is how you have to understand or remember the scientific classification over here. So they are chromatophores, as I told you all. So they have a chromatophore present over here, which helps in photosynthesis. That is, the chromatophores have chlorophyll, which helps in photosynthesis. Nutrition, holophytic, which takes place by phototrophy, that is by photosynthesis, and also holophytic or holozoic. The reserve food is starch, and organisms may have one or two flagella. Example of here is Euglena. So there's a Euglena. Then you have
Your voice is
any disturbance from my side?
No, ma'am, it's clear.
Clear?
Clear, clear.
And then you have Noctiluca. So this is a Noctiluca over there. And so these are some examples of pictures I put it for you all. And coming to Zoomastigophora. Under Zoomastigophora, that is zoo means animal. Let me just close the door and come 1 second. There's some noise from my side too. Disturbance. Yeah, Zoomastigophora. So these organisms do not have chlorophyll. So that is the reason why they are called as animals, because they don't have chlorophyll, means they are unable to prepare its own food. So they have to depend on the plants for their food. Mostly parasitic. Nutrition in these organisms, holozoic or saprozoic, and the reserve food is glycogen. And they may have one or many flagella. So you can find they may have one or many flagella. Examples are Leishmania over here. So this is a Leishmania with a single flagella. Then you have a Trypanosoma over here with a single flagella. Trichomonas with many flagellas, and Trichonympha over here. So these are the examples over here in Zoomastigophora. And under these two, the Leishmania and the Trypanosoma are the ones we are going to talk about in the further classes. Then coming to Opalinata, as I told you, Opalinata doesn't have a class. So this belongs to superclass, or which is they live in commensals, or that is commensals means mutualistic. or they are parasites in the gut of anurans. What is anurans? The frogs are called as anurans, and mostly Opalinata infects the frog. Body is covered by oblique rows of cilia like flagella. So here the cilia, they look like cilia, but they're not cilia. They are called as flagella. So that's why they're called cilia like flagella. And the organisms may have 2 or many nuclei. So you can see there are so many nuclei in this Opalina, and in Zillerella, you have only 2 nucleus over there. So 2 or many nucleus. And they undergo reproduction by binary fission or by syngamy. What is syngamy? Exchange of gametes is called as syngamy. And sexual reproduction takes place by anisogamy. So you have different terminologies over isogamy and anisogamy under syngamy. So what you call as syngamy, exchange of gametes is called syngamy. So when you're talking about anisogamy, the gametes are of 2 different sizes. That is, the male gametes are smaller and the female gametes are larger ones. The eggs are large and the sperms are small ones. So that is called as anisogamy. When you're talking about only isogamy, isogamy is when both the gametes are of the same size. There is no difference in the size. So that's called isogamy. What is the difference now? I hope you understand. Aniso means the size of the gametes are different in anisogamy. And when you're talking about isogamy, the size of the gametes are the same ones. And coming to superclass Sarcoidina. Sarcoidina, as I told you, is fleshy. The locomotion is brought about by pseudopodia. Their body is amoeboid with definite pellicle. So the body is covered by definite pellicle. Nutrition is holozoic or saprozoic. That is by diffusion, saprozoic. And this is again divided into 3 important classes. Rhizopoda. Rhizo means, I told you, root. Poda means foot. And they form lobopodia, philopodia, or reticulopodia. Means what? These are the different types of foot that you find in this type of rhizopodia. It can be lobo. Lobo is thick lobe-like structure foot. Philopodia is a filament-like podia. And reticulopodia, you can see those thin elongated lines over here. So these are called the reticulopodia in Lphdm. So this is an example of an Lphdm with axial filament. And these include amoebas, foraminiferans, and mycetozoans. And they are free living and also parasitic. And the body is always covered by porous calcareous shell. So these are always covered by calcareous. Calcareous means deposition of calcium is called as calcareous shell. Example, amoeba, entamoeba, and Lphdm. Then coming to the second class, pyroplasma, pear shape. If you can see, this is the RBCs over here. And in one RBC, you can find those pear-shaped pyroplasmia, that is babesia. So this is one parasite that is inhabiting the RBCs over here. So as I told you, because they are spores, so they form a spore-like structures. And locomotor structures in the spores are absent. And they are very small particles that are found in the RBCs. Example, babesia. And the next one is actinopoda. Actino means, I told you, ray-like structures. Can you see the picture over here? They resemble the rays over here. So this is called a ray-like structure. So that is why they come under actinopodia. Examples are Colosseum, Actinophis, and Actinometra. And they have this axopodia with axial filaments radiating from the spherical body. They are planktonic, and they include helizoans, radiolarians, and acantharians. So these are mostly you find in the ocean after they are dead. They form a radiolarian ooze. What you call them as oozes. You will find the whole ocean bed covered by this particular siliceous shells that is found under the marine sea water or oceans. That is why they are called as radiolarian ooze. I can show you this picture over here. This is how the picture looks like under the bottom of the ocean over here. So you can see, as I told you, because they are calcareous, made up of calcium deposits, you don't find them this big. I wrote over here, this is under microscope at 250 magnification. So you have to view them under microscope. Why? Because this radiolarians are almost just 1 mm to 2 mm in size. They are very, very minute. Mm is 0.0 centimeters. So when I'm talking about 1 mm, they are almost 0.1 centimeter large. So you'll have to definitely view them under the microscope. So what exactly is radiolarian ooze under the bottom of the sea is what this picture looks like. Just for your info over there, I told you. Then coming to the next subphylum Sporozoa, endoparasites mostly. The sporozoans are endoparasite. Endo means inside, ecto means outside. So these are all inside the body, endoparasites. Special locomotory organs are absent in them because they are spore shaped. So special locomotory organs are absent. Sometimes they have pseudopodia, which helps them also in ingestion of the food. And merozoite is a stage which has an apical complex. That is, they have an apical complex like this, the merozoites, which helps in penetrating into the RBCs. So that is the only type of foot that they have. That is an apical complex, which helps in penetrating into the host cell. And they are divided into 3 classes. What are they? They have the telospora. The sporozoites are long in these animals. Reproduction is both by asexual and sexual reproduction. And these are the parasites of the blood and the gut. And reproduction over here is by isogamy or anisogamy. So now you, I hope you understand what is the difference between isogamy and anisogamy. The examples
Yes,
over
ma'am.
here, the example over here is monocystis, then Imera and Plasmodium. So these are the examples over here under Telospora. Now coming to the next one, Toxoplasmia. Toxoplasmia over here, these are the parasites, Toxoplasma examples over here, which is having a elliptical shape over here, and they produce by asexual reproduction. Sometimes budding is found where 2 daughter cells are produced within the mother cell, and finally the mother is destroyed, and spores are absent over here. And example over here is Toxoplasma and Haplosporia. Amoeboid-like structures over here, so you can find the small amoeboid-like haplosporians. Reproduction is only through asexual, that is by multiple fission. That is, one single cell divides into many number of cells. So multiple fission occurs over here. And example over here is Haplosporidium over here and Ichthyosporidium. Now this Ichthyosporidium you will come across when you're talking about disease in the fishes. They attack the fishes mostly, and they just hang on to the flesh of the fish, and they create a sort of itching sensation to the fishes. That is why itching. Ichtyo is itching. Ichthyosporidium. Just an example over there, what exactly they do. And coming to subphylum Nidospora, as I told, needle, spore over here is seed. So it has some needles in the seed-like structures. That is what it looks like. So these are the polar filaments. Some have 2 polar filaments. Some animals have only single polar filament over here. With the help of this, they are able to move and do different types of activities. They are parasitic, and as I said, they have 2 or more filaments. And these polar filaments are unique feature in these animals, and these spores infest a host. And then the polar filament is discharged and it gets attached to the host tissue. And they are of 2 types, Myxosporidia, and you have another one, Microsporidia. The spores of this animal of this class are large and develop from several nuclei, and they are generally extracellular parasites, and the spores of this class have 2 polar filaments. So that is what is the example over here. And Microsporidia, you have only 1 single polar filament over here, and these are also intracellular parasites. And example over here is Nosema bombycis. So that is one example over here. And coming to the last one, subphylum, that is Coeliophora, cilium, that is eyelids with lashes, and forus is bearing. That is, they have those lashes-like structures. That is, they have so many cilia around them. That is why they're called as lashes of the eyes. So Coeliophora, or the presence of cilia on them. They are complex protozoans, and locomotory organs of this subphylum is cilia. That is why it's called Coeliophora. And the nuclei of these organisms are dimorphic. Dimorphic is 2 types. They either have macronucleus And they also have a micronucleus. The macronucleus helps in vegetative and polyploidy, whereas the micronucleus helps in reproduction. And reproduction takes place by binary fission, and sometimes sexual reproduction takes place by conjugation. And they have only one class, that is ciliata. And under ciliata, you have the examples of your Paramecium. You can find, we have been studying Paramecium from 10 inter, I suppose. So you have those cilia present over them. And I was talking about this macronucleus and a micronucleus of that small one over there, micronucleus. Then they have those food vacuoles, and they also have the chlorophyll present, not the chlorophyll, sorry. So they have the food vacuoles for the consumption of food over there. And they have these radiating canals where the contractile vacuole is, the radiating canal takes in the food, and then the contractile vacuole helps in digesting the food. Then you have a vorticella over here, that is a miniature tulip-like structures, flower-like structures that you find. The cilia is present on top over there, so vorticella. And Lphdm is another example of here with complete cilia present in them. So this is about the class ciliata. And so this was the classification of protozoans. And we talked about 4 subphylum. And I would like to take for another 10 minutes. Is it okay with you all students?
Okay, ma'am.
Okay, ma'am.
Okay, ma'am. We'll
Okay, ma'am.
complete this type study, and then in the next class, we'll talk about the diseases of it. So Lphdm is a very beautiful protozoan because you will come to know some important characters about this Lphdm. Can anybody tell me to which class they belong to, Lphdm?
Sarcoidina.
Sarcoidina?
Rhizopodia.
Rhizopoda.
ma'am.
It's over there. It's over there. So phylum Protozoa, class Rhizopoda, order Foraminifera. Those who were talking, it was superclass. Superclass is Sarcoidina. But I didn't put superclass on there. I directly asked which class they belong to. Yeah, so they are Rhizopoda. Okay. Now, if you can see the picture over here, this is also a calcareous one over here, the fossilized Lphdm over here, and you can find that they have a V-shaped chambers over there. The whole animal is covered by V-shaped chambers. So this is what we are going to talk about in the type study of Lphdm. So what are these chambers and what are these pores that are present in them is what we are going to talk about. And we are also going to talk about the center part, which is a differentiating character. That is, as I told you all, Lphdm is dimorphic in nature. So what is that dimorphism and how they are being studied? Okay. Coming to the type study, Lphdm. This is also a favorite question for the examiners. They will ask you to write. You might get it in a essay writing question, that is for maybe 10 marks. I don't know which one is that. They will ask you to write, discuss about type study of Lphdm. So you're supposed to write the classification. You're supposed to write the classification, and then you're going to start writing about Lphdm, or another name for Lphdm is Polystomela. Okay. Coming to its characters and structures, these are marine forms. So you find them only in the marine waters. Marine means sea, water. Found creeping on the seaweeds to a depth of 1800 feet. So you don't find them on the top waters. You will find them only on the seabeds, where you can find them creeping on the seaweeds. You know what a seaweeds are? Weeds means unwanted poisonous plants. So you can find them creeping around the seaweeds at a depth of 1800 feet. And what is the structure? This is also called as polystomela. Poly means many chambers is polystomela. And it's a dimorphic rhizopod. Why dimorphic? I'll be talking about that. There are 2 types of Lphdm, and they are unicellular, microscopic. And if you can see the size over here, it seems to be too big. But when you're talking about the size, it's only 1 mm in diameter. Okay, did you get that? So from this point to this point, it's only 1 mm in diameter. So you can imagine how small this particular Lphdm is. So as I told you, 1 mm is how much? 0.1 centimeter. It is pale
Yes,
yellow
ma'am.
in color. So they are pale yellow in color, and the body is covered by a biconvex shell. So this is a biconvex shell that is covered, and the first formed chamber is a proloculum. The center part of this particular picture over here is a proloculum, and this is the first formed chamber in the Lphdm, and that is called as the proloculum. This proloculum is the one that differentiates between two different ones, that is a micro and a macro ones. The shell contains spirally arranged V-shaped chambers, hence it is called as polythalamus or multilocular. Okay, so that explains why you call them Lphdm as polystomella, polythalamus or many chambers, and they overlap one above the other, and only the latest one can be seen very clearly. The rest of all of them are fused behind one another. And these radiating ones, what do you call these radiating lines as? I told you all about this. What is that? What is this radiating? What podia you call them as?
Reticulopodia.
Very good, very good. Reticulopodia. Yes, exactly. So that's a
Reticulopodia.
reticulopodia. Yeah. So that's how we are going to discuss as you're studying over here. Now, these chambers show perforations. So you have that small perforations over here. Can you see those perforations, those pore-like structures over there?
Yes, ma'am.
They have those small perforations. The cytoplasm of the whole body of the animal is not divided into compartments. So the whole cytoplasm is dispersed. They don't have compartments. Everything is openly present in this particular Lphdm. So they you don't get confused with these chambers and the cytoplasm, thinking that they are separated from one another, but they are all together. So the cytoplasm is not divided at all, and they don't have they are not separated in the compartments, but they are only continuous. And the cytoplasm extends through the spores of the shell to form a layer over the shell. So one shell is formed, that is a proloculum, and you have another shell over it, and it keeps on adding like that. And this is the last shell that has been formed on this Lphdm. So you can clearly see only the last one, and the rest of them are fused among one another. The cytoplasm is produced into a number of reticulopodia. I told you all what a reticulopodia is, which will form a network. And from the hinder end, you can see this hinder end. So this teeth-like structures. So this teeth-like structure are also called as retral process. They are called as retral process. And cytoplasm contains one or many nuclei. So one or many nuclei is in the macro type of Lphdm. That is when the proloculum is big, so you call this as macro. And if the proloculum is small, you call them as micro over there. The macro one has only one nuclei, whereas the micro Lphdm has many nuclei. So that is the difference again in these two type of Lphdm. Cytoplasm contains food vacuoles, which it takes up the process of digestion. So here you have to remember, food vacuole means they help in digestion of the food that has been ingested by phagocytosis. Or here in the Lphdm, the reticulopodia is the one that captures the food for its nutrition. Okay. So dimorphism, as I told you all, the two type, megalospheric, microspheric type of Lphdm is found over here. So the Lphdm exhibits dimorphism, that is, they occur into two distinct forms, megalospheric and microspheric. So what is a megalospheric? If the proloculum is big in size, so you can see in this picture, if the proloculum is big in size with a single nucleus present, so it has a nucleus over here, single nucleus present, and this takes up sexual reproduction, that means it is called as megalospheric Lphdm, whereas a microspheric Lphdm, the proloculum is very small, you find them very small, and they have many nucleus. Can you see those small dot ones all around the Lphdm? So they have many nuclei present in the cytoplasm, and they reproduce by asexual reproduction. So this is what you'll have to talk about when you are talking about the Lphdm. Main thing you have to talk is megalospheric and microspheric form of Lphdm and how the chambers are being formed and what is the shape of the chambers, V-shaped chambers. So this is some information that I gave you all for you all to undergo, read it so that it will be easy for you all to differentiate between the macro and the microspheric individually. Locomotion over here, locomotion and nutrition. They show slow creeping movements with the help of reticulopodia. So this radiating structures you find. So this is reticulopodia. And from the picture itself, you should be able to tell me whether it is macro or microspheric. Which one is this? Anyone? Can anyone tell me? Is it macrospheric or microspheric?
Micro.
Very good.
Micro.
Very good.
Micro.
Microspheric. Yeah, because you find all the small
Yes, number of nucleus.
number of nucleus and the proloculum is very small. Can you see that?
Yes.
Because macro
Yes.
have been one single big chamber over there. Okay. So you can see over here the reticulopodia is capturing the food over here. Can you see? This is the food over there, and the reticulopodia helps in capturing the food. The reticulopodia digests them outside itself, and then the food is ingested into the cytoplasm. So they are holozoic feeders. That is, they're going to take up the parasite and they're going to put them inside. So this reticulopodia will capture it, and when they come in contact, they secrete a toxic substances which makes the parasite, the food particles to paralyze for a second. And by that time, the food is digested in the food vacuole and the digested food is absorbed by the cytoplasm. So that is locomotion and nutrition. So the reticulopodia helps in locomotion and also in nutrition. That is, they capture the food and they release a toxic substance over them, paralyzing the food particle. So this food particle can be even more tiny microorganisms, or it can be an algae or anything it can be. So they are just going to kill them and they're going to absorb them inside the cytoplasm. So that is locomotion. Coming to reproduction, this is most important topic over here. They might also ask you in a short answers, explain about the reproduction and life cycle of Lphdm. So you're going to just talk about the reproduction and the life cycle of Lphdm in a short answer, and if they ask you in a long answer, then you'll have to talk completely about its structures, locomotion, digestion, and reproduction. So coming to the short part of it, reproduction, they exhibit alteration of generation. So what do you mean by alteration of generation? That is, asexual reproduction follows by sexual reproduction. That is alteration of generation. So the megalospheric form alternate with the microspheric form. And microspheric form always develops by conjugation. That is, we talked about this. Microspheric forms always reproduces asexually, whereas megalospheric form reproduces sexually. So this microspheric form developed by conjugation or syngamy, and megalospheric forms or develops with the conjugation or again a syngamy. What is the difference over here? We will be talking about this. The microspheric form reproduces asexually by fission to produce a number of amoebulae, and the inner cytoplasmic mass containing several nuclei creeps out of the shell and remains as a lump around it, and a small amount of cytoplasm collects around each nucleus. I will show you all in the picture. You will not be understanding it. Now coming to this microspheric form. So this is a microspheric form. And as I told you all, the microspheric form has many nuclei. Yes. All these nuclei separates out from the animal and they come outside. Once they come outside this covering on or the proloculum over here, the cytoplasm also oozes out and forms a membrane around this particular nucleus. Now, all this nuclei, which is present in the microspheric form, will come out, and a little bit of cytoplasm covers around them, and this is called as amoebulae. Okay, so this is called as amoebulae. And this amoebulae, what happens is slowly. Yes, ma'am. Any question? Right. So this amoebulae, what is happening to this amoebulae? First proloculum starts to add on the first proloculum. Then this is the second proloculum, this is the third proloculum. So as and when it is growing, a new proloculum keeps on adding, and this young Lphdm forms into a megalospheric form. So this is a megalospheric form. So this is dividing by asexual reproduction. The nuclei from the mother cells oozes out, and once the nuclei comes out, the cytoplasm also oozes out, forms a protective covering around this nuclei, and this is called amoebulae or amoeboid structures. And once this amoeboid structure starts to grow, the first proloculum is added on, and then followed by different proloculums over here, the surrounding proloculum, and they ultimately becomes a megalospheric form. So it has one large chamber in the center, and it will have only one single nucleus. So this is called a megalospheric form. So from microspheric form, they are changing into megalospheric form. Understood this point? Now coming to megalospheric form, how do this megalospheric form reproduces? Now, as I said again, this reproduction is by asexual reproduction. Microspheric, asexual reproduction. Megalospheric, sexual reproduction. So what is happening over here? Now, in this point, what does a megalospheric form has? Only one single nuclei, I told you. Now, this one single nuclei is going to divide multiple times. So they're going to divide multiple times. And once this nuclei is divided into two or into multiple times, again, each nuclei is going to again divide into a smaller nuclei. And once this smaller nuclei are formed, cytoplasm is surrounding by them, and then they are forming filament-like structures. There's two filament-like structures forming over here, and these are all tiny gametes. They form tiny gametes. Unlike the nuclei over here, wherein it is forming an amoebula, here a single nucleus is going to divide multiple times to form small nuclei, and this small nuclei is going to grow and develop filament-like structures. Once the filaments are formed, they are released to the sea. And once they are released into the sea, this microgametes will search for another microgamete from a different megalospheric form. Got the point? They are not going to conjugate between themselves. They are not going to fuse from the same megalospheric form. This microgametes are going to go and search another microgamete from another megalospheric form, and those two microgametes, they are going to fuse to form a zygote, and this zygote forms into a microspheric form. Follow this point. Any doubts over there? Shall I repeat it once again?
Yeah, ma'am, please. Yeah, ma'am, please.
Fine, fine. So what is happening over here? I'll show you another beautiful picture. So this is what I was explaining to you all. I'm not going to go through this. Okay, coming to this picture, you will understand what exactly this picture is over here. So here, what you are finding over here is, what is this over here? This is a this is a gamete over here, and this is the megalospheric form. Okay, so this megalospheric form over here, the nucleus divides multiple times, and once this is dividing multiple times, what is happening is they are going to give rise to many number of gametes. A gamete, how do you differentiate between a gamete and an amoebula over here? Is that is, when an amoebula is formed, you will find only the nucleus and a cytoplasm surrounding them. Clear? But when a gamete is formed, it is having those filament-like structures, two filaments. Can you see the difference over here? The amoebula over there and the filaments that are present in the gametes. So single nucleus divides multiple times to form into small, minute nucleus, and this nucleus grows to form gametes with a filament. And once these filaments are, the gametes are released into the open sea, each gamete from different parents. So this is one parent. You will have to have another parent over here, and the gametes of that parent is both going to fuse to form a zygote. And this zygote ultimately forms a microspheric form. As they are adding on the proloculum, they form a microspheric form. Now, what is happening in the microspheric form? Once the nuclei comes out. They have numerous number of nucleus. Once the nuclei comes out, the cytoplasm also oozes out and covers the amoebulae over here. So these are the amoebulae, and this amoebulae.
Madam, sorry to disturb you.
Yeah.
Madam, please continue tomorrow.